I cannot share my screen for some reason. Can you allow me screen? Yes, here it is. Okay, thank you. And is it fine? Can you see my screen?
Yes.
Okay, thank you. First of all, thank you very much for the invitation. It's a pleasure to be participating this event. And I want to tell you a little bit what we've done recently in fluid dynamics in the context of biophysics, and in particular in the context of developmental biology, the development of the fly. But the story starts much earlier. It starts in the 18th century with my compatriot Bonaventura Corti. He was an abbot, but he was very curious about nature, and he was in fact the teacher of Venturi, the Venturi effect in fluid dynamics, the drop in pressure when you have a constriction in a tube. And he was using a microscope, and which been around for over a century, so that was no particular novelty. But he used it to observe what was going on in what he called an aquatic plant. In fact, it's an alga, but it doesn't really matter for today's talk. The point is that he was observing this long plant, which about 10 centimeters, that has cylindrical cells extending very high. In 10 centimeters, it's one of the biggest cells that you can have in nature. And he was observing what was going on. There's clearly a problem to bring stuff from the nucleus to the end of the cell. And he was observing what you see on the right. On the right, you see this flow of material which goes into these long cylinders. And those of you, of course, you go back to the fluid dynamics textbook, you remember Poiseuille flow. Poiseuille flow, you remember it's parabolic, and the no-slip boundary conditions, they tell you that close to the walls, you have to have zero velocity. While he was observing, of course, he didn't know about Poiseuille flow, but he was observing this fascinating effect that you have these two parallel streams going on into the cylinder. Nowadays, we recognize that this cannot be obtained by a passive flow. It must necessarily involve some active mechanisms. And that's the charm, and that's the beauty of this fluid dynamics in biology, that very often you have some active mechanism, you have some consumption of energy, you have coupling to other effects, like, for example, biochemistry. And this generates, usually they are relatively low Reynolds number flows, but they have quite a complicated nonlinear dynamics, as I'll try to show you later on in the case of development, because of this activity and because of this coupling, because of non-trivial boundary conditions that are present in the problem. Now, just to show you that not everything is active, but let me show you another example of cytoplasmic flows, which happen in the slime mold Physarum. This is work done by Karen Lim and collaborators. The slime mold is it's a very long mold that creates an intricate network, as you can see here. And it features an effect which is very much reminiscent of the peristaltic contractions that we do have ourselves as well. And this is the mechanism that allows the slime mold to propagate, again, stuff. The problem to be solved is that you want to send stuff far away. And clearly diffusion is not a very effective mechanism because it goes like square root of time and it takes too long. So you want to have some advection. And the problem is how to generate advection. In the previous case, you've seen that this is generated by some active mechanism. In this case, the flow itself, of course, it's active as well in the sense that you have contractions, which is shown in the plots here that you have. You see that it periodically contracts, but the flow inside it's a relatively standard Stokes flow, as you see from the equation, which is here. This is the standard Poiseuille flow in the parabolic in a Stokes flow. And what the authors of this paper show is that this kind of modeling with the appropriate contraction of the boundaries, it's able to capture the dynamics of the slime mold physarum behavior. Now, if you want to know more about the stuff that there's a review paper giving a physical perspective on this cytoplasmic streaming by Ray Goldstein and his collaborator, and you can read there. What I want to do today is to show you a little bit about the coupling with biochemistry that you have in developmental biology, and show you a bit how the boundary conditions, they intervene and they can generate quite a non-trivial dynamics in the case of the fly embryo. So let me give you a 101 introduction, very short, to the what Drosophila. Drosophila is the fly, is what you meet, is the fruit fly. So it's what you meet when you leave your fruits for a few days on the desk, on a table. You will see these little flies all around, and they develop in a it's one of the model organisms of preference because it reproduces very fast, and it contains a lot of the mechanisms that are also present in other contexts. The way the development goes on is summarized in this scheme here. Initially, you have a single nucleus. This single nucleus is getting fertilized, and that's something that I will not discuss today. But the fertilization process ends up with a calcium wave. This calcium waves is the signal that triggers the start of divisions of the nuclei. The initial nucleus then starts to have a series of 13 cycles that are summarized here. The green is the S phase, so it's the phase where you synthesize the chromosomes, you copy these chromosomes, and then in red you have the duration of mitosis, so the division, the two chromosomes that were copied, now they get separated, they get split. And this way you go from a single nucleus to of the order of thousands of nuclei. In a time scale, the pace of these nuclear divisions are absolutely frantic for mammalians, because they divide in order of a few minutes, 8 or 9 minutes, just compared to the hours of days that can take divisions in ourselves, in humans. And they divide, and there are several problems that must be overcome in order to achieve a good share of the nuclei. First of all, you don't want all the nuclei to stand at a single location. Typically, the initial nucleus is somewhere. This will be the future head, this will be the future tail, this is the ventral part, and this is the dorsal part that you have here. You wanna bring the nuclei to the whole embryo, and then these nuclei, they move from the bulk of the embryo to the cortex. Once they are at the cortex, they keep dividing until they start posing at cycle 14, and this is the phase of gastrulation. So to make a long story short, let me show you a movie, which is probably much better than what I've been telling you so far. So let me show you how this happens. What you see is the embryo of Drosophila. Here, it's 500 microns. It's a couple of 150 microns on this direction. Again, this is the dorsal part. This is the ventral part. This is the head, future head, and this is the future tail. And what you see is the external part. So it's the cortex of the embryo. And initially, you won't see much other than this movement of the yaw of the cortex that is going on. And then at a certain point, you see, this is the phase where the nuclei are still inside. And now you see the nuclei that have come to the cortex, and they keep dividing, as you see here. These are waves. These are waves of division that we've studied. There's a lot of nice results that we obtained, but I won't have time to discuss today, so that's not the the subject of my today's talk. I'd be happy to discuss with those of you who are interested. This is the phase 14, where the embryo poses, and here it's cellular, and now here's the phase so-called gastrulation, where you create the 3 layers of our body, and you have the creation of the internal organs. So that's the movie. Now, what I'll focus on today is this phase here of the axial expansion, which is the phase where there's a few tens of nuclei. Initially, they're all present in the middle part of the embryo. And the problem that the embryo has to face is that it has to bring these nuclei, distribute them more or less homogeneously over the entire embryo. And this happens at between cycle 4 and cycle 7, which is here. Now, I forgot to mention that the whole process takes about an hour and a half at room temperature. If you cool the temperature of the lab, then the time scale lengthens. And if you make all this process happen at about 10 degrees, then this can take, like, rather than an hour and a half, it can take several tens of hours. So there's a slowdown of the entire process, but everything happens properly and the embryo is still able to develop properly. The reason how this is achieved is not entirely clear. It's a fascinating problem which is still quite open. So let me explain how the embryo manages to achieve this distribution of nuclei more or less homogeneously over the embryo. And the process is shown here. That's a different movie. Now, what you will see is something else. What you will see is that there's been a PCNA that has been stained with red fluorescent protein. What PCNA is, it's a clump that goes around the 2 strands of DNA that are getting copied. So when the S phase is going on and you're copying the strands of DNA, then you will see some signal, some red signal appearing inside the embryo, and then it will disappear. The fact that it disappeared doesn't mean that the protein is not there anymore. It just means that the clump is not attached to the DNA because the DNA is not getting copied. So let me show you the movie, and you will see the nuclei. And you see these nuclei that are moving very rapidly, order of microns per second. And you see that by the end of this process, all these nuclei that were initially concentrated around here, they have multiplied and they've distributed all over the embryo. Okay, so how is this achieved by Drosophila? First of all, let me show you these trajectories, the trajectories of the particles that you can put inside the embryo, how they, what kind of trajectories of Lagrangian trajectories they draw, and you see that you have a kind of 4-vortex flow. Like here, so there's a flow which goes in this direction here, then it goes inside. There's a stagnation point more or less around here at about 40% of the embryo length. And then there's this expansion that you have in the 2 directions that is getting produced. So this is the kind of flows that we want to explain and want to capture. And the first temptation, of course, is to do like it was done in the previous, in most of previous works, which is to say that the flow and the bulk, the cytoplasm, what is inside the embryo, is in fact a Stokes fluid. A Stokes fluid, it's passive, of course, by definition. And so what you can do is that you can take the boundary conditions, which is the flow close to the cortex. Now, of course, the fluid dynamicist will object to this, and I will come back to this because a passive fluid cannot slide onto a still surface because the cortex doesn't move. Therefore, we are violating somehow the no-slip boundary conditions. But let's forget for a second about this. Let's take violate the no-slip boundary conditions, take from the experiments the the flow close to the cortex, and then calculate the Stokes flow, the corresponding Stokes flow inside the embryo. Now, if you look at this, you could conclude that after all, the large-scale flow is relatively well captured, at least the large-scale dynamics. In reality, the agreement, which visually looks okay, it's in fact quite unsatisfactory. First of all, because you miss some of the features at the small scales, and the best way to show this is to take a plot of the vorticity. The vorticity is the rotational of the velocity, and therefore, if you take a Stokes flow, Laplacian of V proportional to grad of pressure, you take the curl of this equation, and you conclude that the Laplacian of omega is equal to 0. Laplacian of omega equal to 0, it means I forgot to say that most of these flows are two-dimensional because they happen in the plane of the, we are taking the mid-plane, so there's no reason for the flow to go left or right in either direction. This is just for simplicity, to simplify the presentation. Laplacian of omega equal to 0, it means that omega is harmonic. Harmonic functions don't take extrema inside the domain. They can only have extrema at the boundaries. And so you can go and check very easily in a very simple but very effective way whether this is a Stokes flow or not by by measuring the vorticity and checking whether or not the maxima and minima are inside or at the boundary. And as you can see from this plot here, obtained by my collaborators and friends in Duke University at the cell biology department, the extrema of vorticity are inside, and therefore the flow cannot be Stokes. Furthermore, the other reason why it's unsatisfactory to consider this a Stokes flow is that what I was mentioning, which is the boundary conditions at the boundary cannot move. They cannot be motion at the fixed boundary for a Stokes passive flow. So we have to do something else if we want to really understand what is going on. And let me show you a cartoon of what is really going on, and you will see why these flows are in fact so interesting, provided you couple them and you spend a little bit of time talking to biologists. What is really going on is summarized in this cartoon. The nuclei, which are inside the bulk of the embryo, what they do is that they go through a cell cycle. Now, the cell cycle is regulated by some proteins. In particular, there's a regulator of the cell cycle, which is called CDK1. Now, this CDK1, the name of this molecule is not so important, but just think of it as a clock. The level of this protein goes up and down periodically with the cell cycle. When the level of CDK1 is low, is the beginning of the cell cycle. When the level of CDK1 is high, then it's the end of the cell cycle, and the chromosomes, they divide. Now, as the cell cycle goes on, there's another protein. There's a phosphatase. For those of you who are in biology, the CDK1, it's a kinase. This phosphatase PP1 regulates the activity of CDK1 and brings it down at the end of the cell cycle. And it's secreted by the nuclei and creates a sort of a cloud, a spherical cloud of about 50 microns of radius around the nuclei. Okay, so you should imagine this nuclei surrounded by this cloud, which goes about 50, 60 microns in either direction. And you remember the dimensions of the embryo. This makes for touching about the cortex at the locations where the nuclei are present. And there's very low levels around here where the nuclei are not present. Now, this protein, PP1, it's able to regulate the activity, the active part of the embryo, which is the myosin. The myosin is the molecular motor which is present in our muscles and is what is able to contract. So we are able to contract our muscles thanks to the activity of the myosin, which takes the fibers of actin and it's able to pull them together. And by pulling them together, it's able to generate contractions. This is the contraction is summarized by this red lines that you see here. And PP1 regulates the activity of actomyosin by increasing activating myosin, and therefore the strongest contraction, they happen at the location where this cloud of PP1 is touching, and this is where you have the contractions. Now, the contractions, what they do is that they generate what was missing before, which is the sliding along the fluid. Now, the sliding of this actomyosin, now it's able by, you will see momentarily, by friction, to drive the salt, to drive the fluid, the water-like fluid, which is present inside the embryo. And the entrainment that is of this contraction of the fluid, of the sol, generates this 4 vortices that you were seeing before. And this sol now is incompressible. So you have these 2 streams generated by the 2 sides. They clash at the location where most of the nuclei are present. So the only way to preserve incompressibility is to go in. When they go in, they go to the stagnation point. You have another clash of fluid coming from above and below, more or less in the center. And therefore, by incompressibility, it has to go to the anterior posterior. And this generates the extension along the anterior posterior axis that you've seen before, and the extension of the nuclei that you've seen. Okay, so these are words. This was published in a biology paper, but later, more recently, we published the equations. So let me give you a little bit more of the equations. I hope I convinced you by now that you need to have a multi-phase flow, because a single fluid, passive fluid, cannot account for everything. And a purely active fluid is not what you see, because the sol is indeed passive. So what are the equations? Reynolds number is very small. You can estimate it by the viscosity and the typical velocity. The embryo size, it's 100 of microns. So you'll not be surprised to learn that the Reynolds number is low. We can drop out the acceleration and write down a Stokes-like equation, but for 2 fluids now. So you have the divergence of the stress tensor, that includes the dissipative fluid-like part, which is here. So this is the usual passive part of a compressible fluid that you have on this side. And then you have an active part. This active part is accounting for the activity of the actomyosin, which is written here. We take a very simple form of activity, so it's isotropic. I can discuss more details if you want. But the interesting part is then that the stress tensor is equated now to the friction between the gel and the sol. The gel, again, is the actomyosin part, the active one, and the sol is the passive part. And the equation for the passive part is the Stokes that you before, but now on the left-hand side, you don't have 0, you have the friction between the two parts. And then what you do is that you write transport equation for the density of actomyosin, which can be bound or unbound to the filaments of actin. And these are the equations that you integrate. I can give you all the details, but in 25 minutes, I think 30 minutes, I think I'd better go to the results and show you what these equations lead to and give you as results. And what they give you is the following. First of all, you recapitulate the flow pattern. This is simulations. These are simulations of, we start from the cell cycle 3. So you have this 8 nuclei, which are located more or less at 40% of the embryo side, as I was saying before. And this is the way they move. And this is what the flows are. They go up and down in amplitude because of the cell cycle. So the cell cycle modulates this activity. And we can go back to the movie. This is what happens when you have an active fluid And this is what happens when you don't have the active part of the fluid. And clearly you see that the contractions of the myosin is what drives the extension of the fluid. If there's no activity and no contractions, then the fluid, the nuclei, they essentially stay inside here in a relatively restricted region. They push onto each other by a different mechanism, which is microtubule, but they don't extend over the embryo. And I don't show plots here, but if you don't allow for the actomyosin and you do the experiments in the embryo, then you would see that there's major defects that are getting generated in the development of the fly, and the fly will in fact die if you suppress the extension. So it's not just for beauty that the animal is doing this. It's because otherwise it doesn't develop properly. As you can imagine, the nuclei must be distributed all over in order then to create all the organs at the right location and to make the entire body populated with cells. Now, these are the plots of the fluid flow, a snapshot of what the fluid flow looks like. So this is the sole, and you can see from this, and this is the gel. And you can clearly see that having two fluids is not just for fluids, it's not just for beauty, and it's not just to make a good title with two fluid flows. It's really that the two phases of the dynamics, the sole and the gel, they move in a genuinely different way. And in particular, you have these four vortices which are created for the sole, and you have a very different pattern of motion for the gel, which tends to flow to the cortex during the cell cycle. So in spite of the friction which is present between the two fluids, you have the possibility of differential motion between the two phases. Now, I have to say that experimentally, the sole is easily visible. The gel is not easily visible. So the experiments that I've reported before were just reporting on the sole, and the experiment on the gel are on the way, but we don't have the gel motion yet available. Now, I was mentioning that there's friction. If you have two fluids moving one with respect to the other, there's going to be a boundary layer because they strongly move with respect to each other. And in particular, of course, where the boundary layer, there's no turbulent boundary layer in this case because there's no turbulence, there's no high Reynolds number, but the boundary layer is generated by the friction. It's generated by the friction and again by the boundary conditions. In low Reynolds number, everything is about boundary conditions. So what happens close to the cortex? Close to the cortex, the gel, because it's active, it's able to contract and it's able to slide along the fluid because it's consuming energy in order to move. So it doesn't need it doesn't respect the no-slip boundary conditions, so it can move. While the passive part, the sole, is not able to move because it has to respect the no-slip boundary conditions. So over there, you clearly expect that the two fluid will move in a very, very different way. And indeed, this is what happens. And if you make a sketchy, very simple argument on the vorticity of the two fluids, you balance the two and you calculate what the difference in the vorticity of the two fluid is going to do. It's obeying a Yukawa type equations with the length scale squared, which the inverse of a length scale squared, which comes from here. So you can estimate what this length scale is. And it comes out to be, with the parameters that we got from the fit to the experiments, we get numbers of the order of 1 or a couple of microns. So the prediction which comes from this analysis, theoretical analysis, is that there should be a boundary layer of the order of 1 or 2 microns. And it's a real prediction in the sense that this is what the numerical simulations, they show. The profile of the velocity inside a cut of the embryo like this. And this is what the experiments so far are able to report. So they don't have the resolution sufficient to get very close to the cortex. They show something which is consistent and compatible with what we predicted. But again, there must be, which is a good, it means that theory is not just rationalizing post dicto what has been observed. It's predicting something which should be experimentally verified, which is the presence of a drop for the soul close to the. So this part is not. It's again underway. Let me conclude with the beauty. I think what I find, I'm fascinated by biology because it's not the first time that I see animals managing to exploit physics for their own purposes. Obviously, they don't read books. They haven't read Landau-Lifshitz fluid dynamics, but they're still able to exploit fluid dynamics for their purpose. And in particular, they managed to use the fluid dynamics that I've sketched before in order to ensure something which obviously is very important to them, which is structural stability. What do I mean by structural stability is the following thing. I told you that typically the initial nucleus, it's about 40% of the length along the anterior-posterior axis, and it's more or less in the middle of the embryo. But of course, this is biology. It's not like it's a fixed location. Stuff is moving, it's going around. It can be 50%, it can be 60% of the length, or it can be 20%. So the nuclei, in fact, the wild type, typically it's at this location. But in other embryos, the nuclei are, for example, at this location. Or we can take an extreme point of view. They can be completely messed up. This rarely happens in embryos, but we can consider this to be present in what would be the posterior. So push the situation very much to the posterior. And clearly, what the embryo should be doing is that it should still be able, if it wants to develop, I mentioned before, that it should distribute the nuclei all over homogeneously. So it should still achieve homogeneity. And this is exactly what the fluid dynamics that I've been telling you before is doing, because if you simulate, that's what you see. So this is the wild type. And you see that in other situations, even though you're starting from very different initial condition, you still manage to achieve the same effect as before. And how is this done? Well, we can go back to the picture that I was showing here, to the scheme. What is going to happen? Well, if the nuclei are present here, then the contractions are going to happen at the location of the nuclei, because this is where the contractions happens. So the fluid is going to be driven here, and it's going to generate these 4 vortices at this location where the nuclei are present. If I push the nuclei around here to the posterior, then the pattern of the flow is going to change. It's going to change because that's where the contractions are being present. So at the same time, the nucleus is passively transported by the flow, but it's what generating the flow. And therefore, if I modify the location of the nuclei, I also modify the geometry of the flow, and I'm going to move around the location. And as you can see from the flows, here is the 4 vortices that you've seen before. These 4 vortices, they get heavily distorted here. You still have a little bit of counterflow. But if you really push to this side, there's essentially a unique flow which is bringing everything to the other side. There's other predictions that you can make. For example, the embryo is usually an ellipsoid. But you can make rounder embryos. So these are mutants that you can obtain. And for rounder embryos, because of the mechanism that I've told you before, you modify the length scale of this on this direction here. So what you would expect is, first of all, that the flows should get more isotropic. The ratio between the Vx anterior posterior and Vy on the other axis should get more around 1, and indeed this is what you see in these experiments here. The other thing that you expect is that because you're elongating in this direction, and therefore there's less of a contraction at the boundary. You should see less of a flow, and indeed this is what you observe in the experiments and in the simulations. They agree with the simulations as well. Now, there's other things that you can read in the paper. In particular, you can read how proteins like morphogens like bicoid are getting transported by these flows, but I think I'm over with time, so let me conclude here. Thank you for your attention, and acknowledge my wonderful collaborators in Duke University for all the experiments that you've seen, and Claudia Hernandez Lopez, who's a PhD student who's been working on all this stuff. And thank you so much.
Thank you very much, Professor Vergassola, for your very interesting lecture. Any question, please? Alexander, thank you very much for a nice lecture. And my question is about cytoplasm. Some time ago, I used to consider cytoplasm as an example of a non-Newtonian fluid. And from your contemporary research, to what extent this point of view could be supported or rejected? Thank you.
Sure. Well, I agree with you 100%. It's non-Newtonian in the. I've shown you that it's an active fluid. So it's clearly you cannot treat it as a purely Newtonian passive fluid. So the point was to be a little bit more quantitative on the way this is non-Newtonian, and in particular, if I want to summarize what I showed you here, is that I showed you that the non-Newtonian nature of the combination of the two fluids is that there's an effective way of treating this based on multi-phase flows. So you can capture the deviation from being non-Newtonian by using this formulation with two fluids, which seems to be quite fruitful, and to capture this dynamics inside the embryo. So short answer is I agree with you 100% that it's non-Newtonian, and what we did was to push a little bit more how non-Newtonian it is.
Thank you. Professor Lopti.
Maybe my question seems to be stupid, but I ask.
There's no stupid.
I know that the two fluid theory in serious superfluidity. What is the difference? Maybe in your case, more correctly say two different fluid theory or model.
Yes. So I'm not sure I got. So your question, can you just repeat exactly what was the question? You said that two fluids, they are different fluids. So I didn't quite hear.
The fluid model is used in theory of superfluidity.
There's two fluids in superfluidity. That's what you mentioned?
Yes, exactly. So the question is, do you speak about two different fluids?
Okay, so in superfluidity, the difference between the two fluids is that one is viscous and the other one is non-viscous. In this case, you have two different fluids in the sense that one of them, both of them are viscous. Both of them are low Reynolds number, but the difference is that one is active and the other one is passive. So one of them is a standard fluid. The other one is active, which means it's able to generate itself its motion. And that's how it can violate the no-slip boundary condition. Of course, I grew up as a fluid dynamicist, so I knew about these two fluids in superfluidity, and it's beautiful. It's beautiful example of two fluids, which was source of inspiration for this. But it's in the details is slightly different. What is the difference between the two fluids? It's not about viscosity. It's about the activity. Thank you for the question.
Thank you. Bogdan Leo.
Yeah, I am Bogdan Leo. I have one or maybe three questions. One question: what properties or kind have the initial structural units? But why we must use the two fluid approach, but not use active matter approach?
Okay. Well, this is active matter. You can call it. If I were someone who grew up not doing fluid dynamics, but I grew up doing active matter and flux, I would be calling this active matter. You'll have a beautiful example in the afternoon. You can call this active matter if you like. This is active because as you see from the symbol here, there's an A which stands for active, and active, it means that the stress tensor, there's an active part of the stress tensor which is able to generate contractions. So apart from the keywords, this is active matter. I've been doing more fluid dynamics and biology, so I like to call it too fluid, but if you want, you can call it active matter, yeah.
Okay, but in active matter approach, we have the hydrodynamic equation, and from this equation, you can obtain the free non-equilibrium free energy, and this free energy gives the equation, this hydrodynamic equation, and other properties which can obtain from this solution.
Sure. No, I mean, you can write down a free energy for this. I didn't use the free energy because I integrated the equations, and they are complicated enough that we didn't find. We obtained from these equations all we wanted and all the phenomenology that you've seen there. You can write down the free energy, and it's clearly out of equilibrium, the system, because it's the active part is out of equilibrium. By the way, maybe the point that I should be stressing is that this is a particular case of the equation that you can write for any active fluid. If you go to the review of Jacques Caux, Jean-François Joanny, and Frank Huliker, you can look at some of these equations, much more general with many more terms. This is a special case where you take that the activity of the fluid, this active part of the fluid, is isotropic. In general, the active part could be non-isotropic, it could be polar, it could be nematic. We took the simplest case of isotropic, non-polar, and non-nematic because, as usual, as physicists, you start from the simplest thing. But all this is active matter. You can use the usual tools. But that's what we wanted to obtain, and maybe I should have put a reference to the more general thing. But this is active matter theory.
Thank you.
Thank you.
Thank you. Thank you. Last question. Ну добре.
Okay, I thought there were already many questions, but I was intrigued by this very thin boundary layer, and Massimo, is this repeated in other biological systems, other cell biological systems? Is this something more general than this one example?
Well, it's a good question. It's a good question. I was surprised. You know my trajectory. I come from high Reynolds number, so I had this prejudice that boundary layers are due to viscosity, but this is due to friction. So I suspect that you're going to have much of this in many other systems because the mechanism is quite general. You have two fluids, one active, one passive. Close to a boundary, they necessarily gonna differ very heavily, and because they entrain each other, this is gonna generate a boundary layer. Now, so thin has to do with the numbers, but I suspect it's very general. But before making very big claims on the generality, first of all, I'd like to see that this is getting confirmed by the experiments. As I stressed here, at this stage, this is still a prediction. The experiments are showing this, and the prediction is this. So it's not confirmed yet, but you're pointing to a very important feature, which is that it should be, I expect it to be very general, yes.
Thank you.
The last question.
Plastic flows are everywhere, so I suspect.
No. So, thank you very much for your lecture,
Thank you.
Professor Vergassola. Thank you very much.